The last group of amino acids are the ones that are basically really two groups, two subgroups, but the general group is charged amino acids. That means the R chain carries a net negative or a net positive charge in it, okay, in the R group. Besides the functional group, the amino and carboxy group that can carry charges as well, the R group itself will carry a charge. So those two subgroups or amino acids that are carry charges are divided into acidic amino acids. That means they are going to carry a negative charge in their R group. So basically, they're going to have a carboxylic group as part of the R group. And for the most part, that carboxylic group will be dissociated in the form of COO minus, and hence the negative charge. That's the minus there. So under most conditions, specifically around neutral pH, the R group will be negatively charged and will impart a negative charge to that amino acid. So there are two of these acidic amino acids, aspartic and glutamic. Aspartic, 2 carbons, has an extra carboxylic group. Okay? Remember, all amino acids will have an amino and a carboxylic group. Well, the R group here has an additional carboxylic group. That carboxylic group will be most likely dissociated at neutral pH, and therefore the net charge here will be minus. That H plus dissociation will give you a COO minus. And when you go to a COO minus, aspartic acid technically is called aspartate. That means the proton has dissociated. Although we are not going to differentiate between the two, but I want you to understand its meaning, we will use aspartic acid and aspartate interchangeably. Similarly, glutamic acid, similar to aspartic, it's a little longer, and 1 extra carbon, but the functional group that gives it a negative charge is a carboxylic group. So this will be dissociated and gives this side chain a negative charge. Okay? So, as I mentioned, when these are dissociated, when the proton is dissociated, it's referred to as aspartate, and similarly for glutamate. But again, they are the same amino acid. It just describes the state. So we will not distinguish that state or differentiate between aspartate or aspartic. We will use them kind of the terms or the names interchangeable. Okay? The last subgroup, if you want, of the charged amino acids are the basic amino acids, or the R group that has a positive net charge. So there are 3 of these. I'm going to start with lysine. And here's the R group of lysine. Pretty long, large R group with a amino end at the epsilon carbon. This is called the epsilon really carbon here. So you have an amino group, and under normal physiological pH, this amino group will be protonated, so it's in the NH3 plus form, and therefore it gives lysine a net positive charge in the side chain. The second one here, arginine, has a guanidinium group in the R chain, this one here, and you notice it is protonated and has a positive net charge in the R group, and that's arginine. Okay? The third basic amino acid is very important amino acid called histidine. And you see histidine has this ring and called the imidazolium ring structure. You don't need to memorize anything, but histidine has this nitrogen in that ring that is protonated. And again, it gives histidine a net positive charge under physiological pH. Now, the importance of histidine physiologically is that if you look at this nitrogen and this proton, you all can hear me? Okay. And you look at its pK2. So let me explain what pK2 is. pK2 is the pH at which this proton is half dissociated. That means the side chain can exist either half of it is in the protonated form and half of it is in the unprotonated form. So, and this pH, if you think pH 6, that's around pH 6. That's very close to pH 7, which is a physiological pH. So what's unique about histidine is that because of that value of pK2 of its side chain, it can either donate or accept a proton around physiological pH, depending on the microenvironment it is in. And hence its important role in really many different proteins and the way these proteins behave physiologically. Okay? So I just want to tell you that histidine is unique because its pK2 of the R chain is very close to the physiological pH, and therefore it has the capacity to either donate or accept protons under physiological conditions. Okay. So I kind of covered that, and again, you don't need to memorize structures. Just know that which ones belong to which group, and the names, the abbreviation, and that's it. Okay, that's the histidine side chain. That's the arginine side chain.